Hi, everyone. So today we are continuing with Chapter 7 and moving on to Part 2, which is going to focus on cell systems. And so if you remember from Part 1, what we really did in Part 1 was learn that there are two different cell types, prokaryotes versus eukaryotes, right? And hopefully you remembered the distinction between the two. And then we learned the parts of each of those cell types, right? Parts of each. All right. Now, today, what I want to focus in on is what's called cell systems. So here's a way to think of cell systems. Just like if you are a organism like us, you can have organs that combine to organ systems in order to achieve a particular function. In the cell, what we have are organelles that work together to form cell systems to achieve a particular function. So let me give you an example. If we're talking about organs and organ systems, let's take digestion for an example. Digestion means that you need your mouth, your esophagus, your stomach, your small and large intestine to all work together in order to break down your food and extract the nutrients and fluids from that food. So those organs combine into an organ system to achieve digestion. Any individual organ in that system, say just the stomach, can't complete that entire process on its own. Well, in eukaryotic cells in particular, what we find is a similar situation with organelles. So for example, if you wanted to make a protein, maybe like a protein hormone, like insulin, and you wanted to make that and you wanted to secrete it and ship it outside the cell, no individual organelle on its own can do that. You're going to need the ribosomes to make the protein. You're going to need the Golgi apparatus to package that protein and to ship it outside of the cell. So those organelles work together in a cell system to achieve the function of secretion. So that's the kind of thing that we're going to talk about today. Now, just to kind of revisit the structure of this chapter, this is the part that we already went over and was on your exam 1, and if you're watching this in the fall of 2020. This is the part that we're going to be covering today. It will be the subject of your, at least part of the subject of your homework assignment. So the three cellular systems that we will be focusing on for this lecture is the nuclear transport system, the endomembrane system, and the cytoskeleton system, okay? So before we get to that, I kind of want to think a little bit about how we got to, because we're going to be focusing on cell systems in eukaryotes. So I kind of want to focus a little bit on how we went from having prokaryotic cells to getting eukaryotic cells. And that answer lies in something called the endosymbiotic theory. All right, so what is the endosymbiotic theory? All right, endo meaning inside. A symbiosis is a close association between two species or two individuals where they're like biologically linked to provide functions for one another. So what the endosymbiotic theory suggests, and I'm just going to draw here and I'll erase it so I can show you guys these things. The endosymbiotic theory suggests that we had two prokaryotic cells at one point. One was probably bigger than the other. And at some point, this one actually engulfed that one because maybe this particular cell could do a job that this one couldn't do, and that they were able to live together in harmony in a symbiotic relationship that might have given rise to the first eukaryotes. Now, where is our evidence for this theory? The evidence for this theory lies in the structure of these two organelle types that we talked a little bit about in our last lecture. Mitochondria, which of course you remember their job is to make ATP. How many times have you heard that mitochondria are the powerhouse of the cell, right? So mitochondria and then chloroplasts, which of course you know their job is to take carbon dioxide and light energy and convert that into stored energy in the form of sugar, right? So this is another way of saying photosynthesis, right? Okay, so how do these guys provide evidence for the endosymbiotic theory? Well, let's look at the mitochondria for just a second. If you look at it, it looks like a weird jelly bean when you see it in drawings of cells. So here you can see the micrograph of it, and here you can see kind of a nice drawing. First thing to notice is that it's double membraned. You have an outer membrane here, right? And an inner membrane here. Well, let's think about what it would look like if one cell engulfed another. It would look something like this, right? Kind of like a little Pac-Man coming along to eat it. And as this thing moved in, you can imagine a situation where you end up with this guy surrounded by a little pinched off membrane from the host guy, right? So, double membrane could be one piece of evidence. So let's write that down. Oop, come on, eraser. There we go. So one piece of evidence might be double membrane. And I'm just going to put double mem. All right. Piece of evidence number 2. It's not pictured on here, but if we could look really closely at this guy, we would see that it has its own DNA. And more importantly, that DNA is in the form of a single circular chromosome. That sounds an awful lot like a prokaryotic cell, doesn't it? Piece of evidence number 3 has to do with ribosomes. Now, what do I mean by that? Well, y'all remember from the earlier part of this lecture that ribosomes, if we're, they kind of hang out in, this is a cell, and we're going to draw a eukaryotic cell, so this is the nucleus, and ribosomes would hang out in the cytosol, either free or attached to a series of membranes called the endoplasmic reticulum, right? All right. Well, in addition to being out here, this mitochondria has its own ribosomes. And what's even more interesting about the fact that it has its own ribosomes, turns out that prokaryotic ribosomes and eukaryotic ribosomes have some minor structural differences from one another. Guess which one the ribosomes of a mitochondria look like? They look like prokaryotic ribosomes. So that's some pretty strong evidence. The last piece of evidence suggesting to us that mitochondria might have arisen through an endosymbiotic relationship is that they, let's see here, they divide independently of the cell. And I'm just going to put divide independently of the cell. What do I mean by that? Well, most of your other organelles get copied during cell division when the mother cell divides into 2. Not so with these guys. These guys divide on their own. So they're almost functioning like little individual independent units inside a larger unit. And so that provides some pretty strong evidence for endosymbiotic theory. Chloroplasts have all the same attributes. They also have a double membrane, as you can see here. So you see an outer membrane, and then you see all of these inner membranes that are arranged into a series of discs that are stacked upon one another, okay? So they are double membraned. They also have a DNA chromosome that is circular and independent from the rest of the mother cell. They have prokaryotic ribosomes, and they also divide independently of the cell. So the endosymbiotic theory is one way of explaining how we might have gone from a prokaryotic set of cells to a eukaryotic set of cells. Now, once we get to eukaryotes, one thing that I want to mention is that particularly when we're talking about multicellular organisms, cell structure is related to cell function. Now, if you're a unicellular organism like an amoeba, then you probably look a lot like, I'm going to go back here, Like this generalized drawing here. You probably have all of those parts that I taught you in the mitochondria and the Golgi apparatus and all that stuff. You might look, because you need all these organelles in order to perform all the functions that a cell needs. But in multicellular organisms, what we do oftentimes is we delegate different functions to different cell groups, okay? So here's an example. If we're talking about the cardiac cells and the muscle cells in a heart, cardiac muscle cells, what the function we've delegated to this group of cells is contracting, right? To pump blood throughout the body. And so these cells are hungry for ATP. If you look at these cells under a microscope, they look very different than the generalized cell drawings I've been showing you. You can see that all of these, if you looked really closely, these are all mitochondria, okay, that are used to generate the massive amounts of ATP that your heart needs to keep contracting over and over again. Let's take a look at this leaf cell here. You can tell this is an old PowerPoint I've drawn on before. All right, so this is a leaf cell. Leaf cell is going to be heavily involved in photosynthesis. Look here, that is a chloroplast. So we're going to see lots of chloroplasts. And look at this big white space. This is a vacuole, which is a storage organelle. And it's probably storing sugar that has been made during photosynthesis, right? Let's look at this guy. This is a pancreatic cell, right? It makes digestive enzymes. So that means that it makes a bunch of proteins, enzymes, for export from the cell to help break down your food. So we're going to see a lot of rough ER and Golgi apparatus. See all these membranes here? So what I mean by cell structure is related to cell function is that you're going to see the organelles that are related to the cell's job be more common, okay, in a cell type. So like one more example I'll give you guys is a fat cell. If you were to draw like a fat cell from your body, its job is to store lipids as fats, right? So you'd have a nucleus and you'd have a few other things. I mean, you would have some of the other stuff like ribosomes and a little bit of the endomembrane system, but the majority of this cell would be taken up with a big old storage space for lipids. Okay, so structure of the cell is related to its job. And so you should kind of be able to guess like what the job of a cell is based off of the organelles that you see. So let's focus in a little more closely on this concept and let's kind of start our conversation about cell systems. And where I want to begin is in thinking about the nucleus and how we get things inside and out of the nucleus. Now, before we start thinking about getting things in and out, let's review the structure of a nucleus. I'm going to kind of draw you a generalized, I'm going to make this an animal cell because it doesn't have a cell wall. And I'm going to draw like a big nucleus for you to see. Okay. Now the nucleus is double membrane. That does, in this case, we actually don't think the nucleus arose via endosymbiotic theory. Why is it doing that? There we go. So it is double membraned, and then studded within this membrane are a bunch of proteins. Stop it. There we go. Are a bunch of proteins called nuclear pore proteins. Okay. So let's focus in here. This is the outside of the nucleus. So we're looking at the cytosol. Okay. Oof, why is it doing that? Stop it. Okay. This is the inside of the nucleus here. This is the double membrane. And this is the nuclear pore, or actually this is what we call the nuclear pore complex, all right? This inner and outer membrane in combination with the nuclear pore complex is actually called the nuclear envelope. So this area here is the nuclear envelope. Now let's think a little bit more about what's inside the nucleus. You guys know that inside the nucleus, what you primarily have is DNA, right? So here are some chromosomes, they're in their unwound, relaxed state. This is DNA. And remember that DNA's job is that it codes for proteins, right? Okay, so DNA's job is that it codes for proteins. And the interesting thing that we're going to come back to in a second is that DNA never ever leaves the nucleus. And that should make good sense to you because this is the overall instruction book for how to make copies of the cell and how to make the proteins for the cell. You don't want to damage the master copy of the instruction book. So we're going to be making Xeroxes, copies of the master instruction book to ship outside of this nucleus in order to make our proteins. So put a pin in that, okay? One other thing that I wanted to mention as part of the structure of the nucleus, you know, we always see nuclei as being these rounded organelles within cells. What helps them to have their rounded shape is they have some proteins. These are actually cytoskeleton proteins that line the outside, or sorry, line the inside of the nuclear envelope. And they help to give it its shape, and that's called the nuclear lamina. And we're actually going to talk about that a little later in this lecture. So what is the function of the nucleus? Well, first and foremost, you guys know that the function of the nucleus is to store that DNA. Okay? But it's got a couple of other functions too. Two others that I really want to focus in on. Hang on, I'm going to pause this recording and see if I can get it to stop. All right, folks, I cannot seem to get it to stop doing that thing, and I don't want to have to restart the recording one more time, so we're just going to have to suffer through a little bit. Okay, now it's frozen entirely. Let's see if I can get it to stop. All right, folks, trying this one more time, see if it'll work a little bit better. Sorry about the glitch. Okay, so I was saying that the nucleus really has three functions. The first one you already knew. It's going to store DNA. But there are two other things that I want you to know about the nucleus. First of all, it also synthesizes RNA. And we're going to talk about that at great length, okay, later on in the course. So remember I said that the DNA stays inside the cell? But proteins are actually, DNA stays inside the nucleus. But the proteins are actually going to be made out here in the cytosol, right? So somehow we have to get a copy of the instructions from inside the nucleus here, out here, to where the proteins are going to be made. So to get the instructions for these proteins from here to here, we make a copy, and that copy is going to be in the form of RNA, specifically messenger RNA. It also synthesizes RNA that are part of ribosomes. So remember that ribosomes, so I'm going to put makes ribosomes. So remember that ribosomes are those protein-making organelles. So they're out here. Their job is to make protein, but they are actually, if you were to dissect a ribosome, it's made up of both RNA and protein components. So it's not just an enzyme. It's a little bit of both of these things, all right? Together, these guys function to make new proteins. Well, the RNA for a ribosome is made in the nucleus, and there it's combined with other proteins. And if you remember, I'm going to erase some of this from our last lecture, there's actually a little region of the nucleus that's often stained more darkly than the rest of the nucleus. So it looks a little bit like a nucleus inside of a nucleus, and it's called the nucleolus. Okay, so this nucleolus is what's responsible for making ribosomes. Overall, three different functions in the nucleus: store DNA, synthesize RNA both as a messenger, mRNA, or as part of the ribosomes, rRNA. And then additionally, it takes, the nucleus makes ribosomes by taking this rRNA and combining it with proteins, okay? Now, let's take a closer look at this figure that I have for you here. So again, we have this nuclear pore complex, and I mentioned that the DNA stays inside the nucleus and can't leave. But there are things that need to get inside and out of the nucleus. Let me erase this. So things that need to leave the nucleus are ribosomes and mRNA. So again, here are our ribosomes out here in the cytosol. If they're manufactured in here, we have to have a way of transporting them out, don't we? Similarly, if we're going to make a messenger RNA molecule, a copy of instructions on how to make a particular protein, and the proteins are made out here in the ribosomes, then we've also got to export that mRNA out of the nucleus, don't we? So things that need to leave the nucleus are ribosomes and mRNA. Things that need to come into the nucleus are the building blocks for DNA and RNA. There are some proteins that need to come into the nucleus. So inbound, we have things like nucleotides that want to come into the nucleus. And we have a whole bunch of proteins that want to come into the nucleus. Proteins that are involved in being added on to our RNA to make ribosomes. Proteins that are involved in copying DNA. All of these proteins need to be brought into the nucleus. What needs to be exported from the nucleus are things like ribosomes, and messenger RNA. Okay? So now we immediately have a little bit of a question set up for us. What controls the entry and exit of stuff from the nucleus? All right, how do we control that? Okay, so that's what really we're going to be talking about for this cell system, is this idea of what's controlling things into and out of the nucleus. In other words, what's controlling nuclear transport. Now, let's talk a little bit about getting things into the cell first, okay? So we're just going to use getting into, I said into the cell, shoot, into the nucleus first. All right. So in thinking about things that need to get into the nucleus, we know that we need nucleotides. And the good news about nucleotides is they are small. So small molecules like nucleotides, they can actually get through this nuclear pore complex through the simple process of diffusion, right? In other words, they can move through here by moving from areas of high concentration to areas of low concentration. So they can just zoop right through here. But what about larger molecules? You know, we found some interesting things when we started studying molecules that first of all, were getting to the nucleus, and then how we could get them through this nuclear pore complex. And one of the interesting things that we found is that you could have two proteins that are the same size, but both might not make it in. What do I mean by that? So you could have protein A and you could have protein B, and maybe they're both something like 50 amino acids long. So they're about the same size, okay? And protein A cannot make it through here, but protein B could. And so what that tells us is that this nuclear pore is not just a size filter, right? There's something else that controls whether or not things are getting into or out of the nucleus. And so that was a question that researchers were asking: how do we know what can get in and what can get out? Is there something about protein B that's different from protein A that allows it to get in? And the answer is yes. Protein B contains something called an NLS, okay? An NLS is going to function like a zip code signal, and I'm going to tell you what NLS stands for in a second. And so what it does is it helps proteins find the nucleus and enter the nucleus. All right, so let me go into a little bit more detail about that. NLS stands for nuclear localization signal. I'm just going to abbreviate that because it's in your notes. So if you have trouble with that, it is right down there for you, okay? And so what is that? It's a sequence of about 17 amino acids on a protein that functions like a tag or a zip code. If a protein has these 17 amino acids, then basically other things bind to it to truck it up to the nuclear pore. Then the nuclear pore complex can recognize this protein as something that's supposed to go into the nucleus. And you can see here, it's kind of being slingshot up into the nucleus. So the nuclear localization signal, you can think of it as a tag or a zip code, if you will, that helps the protein target the nucleus and get in. So let me show you that one more time. Here we have a protein that has a nuclear localization signal. This nuclear localization signal protein is actually recognized by other molecules, these yellow and green molecules here. See how they've all bound together? You can kind of think of these as like truckers that are going to truck a product up to the nuclear pore. Once it reaches the nuclear pore, it binds to some of the nuclear pore complex and it gets slingshot in there. If we remove this nuclear localization signal from this protein, none of this process would occur and this particular protein would not make it into the nucleus, okay? So this brings up the idea of cellular receptors, okay? Cellular receptors are proteins that can recognize specific molecules. Specific molecules. So how does that work in this particular example? Well, once again, let me erase. In this example, the nuclear pore is functioning like a receptor. And this receptor will only recognize specific molecules that have the nuclear localization sequence, okay? So I said, what are the receptors in the diagram? It's the nuclear pores. And here it says, what's NPC? That stands for something called nuclear pore complex. And I think I only put that on there because I used to have a larger diagram on this slide that had that abbreviation on there, nuclear pore. Don't worry too much about that. I think, again, I only had that defined on here because I used to have a larger version of this slide up. All right. So that in a nutshell is how we are able to get things into the nucleus. So how do we get things out of the nucleus? Well, there is a similar mechanism to get things out. It is also a signal sequence, okay? So molecules that need to get out of the nucleus also have a chemical tag. It may or may not be a series of amino acids. It can be sugars or other things, but there's a chemical tag that allows things to exit the nucleus the same way that we have a chemical tag up here with these amino acids that are letting things get into the nucleus, okay? So that's our first cellular system, is nuclear transport, all right? Now let's look at our next cellular system, and that is the endomembrane system here. And the endomembrane system, to me, is the best example of a cellular system where we have multiple organelles working together to perform a function the same way your body has multiple organs working together to perform a function. So let's take a look at this diagram here. Now, the job of the endomembrane system is often to synthesize proteins, or it could be to synthesize lipids. We'll see that in a second. That's a parenthesis here. Rewrite that. Okay, synthesize proteins or lipids, but my example, I'm going to focus on proteins. All right, So its job is to synthesize proteins and then to transport them throughout the cell. And we're going to need a series of organelles in order to do all of those jobs. So how does this work? So here you can see I've got a bunch of little ribosomes, right? Remember, ribosomes, their job is to make proteins. And here you can see that threaded within that ribosome is some RNA. Now, if I were to draw you a ribosome diagram here, it would look like a little bit of a hamburger. It's got two parts to it. The bottom bun of the hamburger is bigger than the top bun. RNA gets threaded through here as the instruction manual. And what this ribosome does is it reads the RNA and it changes this RNA into a protein, okay? Now, typically what starts out happening is our little hamburger is actually loose in the cytosol. And as this process begins, as we begin to go from RNA into a protein, this will then move to the ER, the endoplasmic reticulum, and the ribosomes will stick to the outer surface of the ER. Do y'all remember that there were two forms of ER, the smooth ER and the rough ER? Well, what differentiates smooth from rough endoplasmic reticulum are the presence of these ribosomes on the surface of the endoplasmic reticulum. So you can see here that we have lots of ribosomes on this endoplasmic reticulum, indicating that we have lots of proteins being made, okay? So as this, see this ribosome right here is making a protein. It's using this RNA instruction book to make a protein here. And the protein is being sort of ejected into the interior of the endoplasmic reticulum. So this ER here, this endoplasmic reticulum, is really just a series of membranes. Can you see that it's just a whole bunch of membranes here? So once this protein enters into the endoplasmic reticulum, what happens is it moves through these membranes and a little bit of these membranes pinch off with the protein product inside. This protein product membrane complex is then moved through the cell to the next organelle that's going to be involved, and that is the Golgi apparatus, okay? Here it says cis face of the Golgi apparatus. Cis just means next to something. So the cis face of the Golgi apparatus is just the side of the Golgi apparatus that is closest to the endoplasmic reticulum, okay? The trans face is the part that's away from the ER and is actually usually closer to the cell membrane. So we have this protein product. Now it's moving to the Golgi apparatus. Now what's the job of the Golgi apparatus? Well, I like to think of this as sort of like a factory model. And I think that might help you understand the difference in the function of these two organelles. So the endoplasmic reticulum is making these proteins, and the reason it's making proteins is because we have these ribosomes stuck on the outside, okay? So they're ejecting their protein product in here. So you can think of this as like just sort of a factory that makes a metal item, right? Maybe it's making a piece of jewelry. We'll say it's making a ring. Now, we ship this very simple metal ring to the Golgi apparatus. The Golgi apparatus is going to be doing a lot of like finishing of the protein. And to keep our analogy going, if this was a metal ring, what the Golgi apparatus is doing is polishing it, maybe adding a stone or something like that, engraving or something, adding put decoration, put deco. So the endoplasmic reticulum makes the protein, the Golgi apparatus processes it. It might add some little chemical groups or make a few changes to it. It's kind of like a factory where you're making a metal ring here and you're shipping it to a second location to polish that ring up and add additional decoration. Hopefully my analogy makes sense to you. All right, so what's going to happen next in this system is This protein gets changed, maybe things, chemical groups get added or taken away. And again, we have a little bit of the membrane of this Golgi apparatus that's going to envelop this protein product in something called a vesicle. You'll often hear these called secretory vesicles because they are going to secrete a protein product. Okay, so here we have our secretory vesicle, and the way it secretes a protein product is by this membrane here fusing with our outer cell membrane. And as it does so, it kind of makes an opening that allows our protein product to diffuse out. This process where you have the cell kind of spitting out a product through membrane fusion is called exocytosis. Okay? Let's look at this a little more closely. So we have the situation where we've made a protein, we've added some bells and whistles to it in the Golgi apparatus, and we're shipping it out of the cell through one of these secretory vesicles that's fusing with the plasma membrane, and out it goes. This is exactly how things like pancreatic enzymes are made. Okay, oops, there we go, enzymes are made. Now, what determines where these secreted proteins go? Because sometimes they might leave the cell. Sometimes these secretory vesicles can go to other organelles. Okay. And so what determines where these things go? Well, just like we had a nuclear localization signal, we have other chemical tags. That act as zip codes that tell these vesicles where to go. I'm going to put vesicle direction. All right, let me go to the next slide and I'll come back to this question in just a second. So let's look here. In this figure, we're blowing up the Golgi apparatus. So here's our Golgi apparatus here. Let me go back. Same as it is right here. All right. So, oh, sorry. So Golgi apparatus here. So what we're doing is we're focusing in on this area of the Golgi apparatus. So this is inside the Golgi apparatus, and this is outside of the Golgi apparatus, or the cytosol, okay? Here are all of the little protein products that have been altered and finished off. Maybe some little chemical additions have been added to them. Notice that each of them has a little bit different chemical tag on them. Sometimes these tags are sugars, sometimes these tags are other proteins, but each of these have a little bit different chemical tag on them, so that when these vesicles pinch off, here, here, and here, the chemical tag tells this protein where to go. So in this example, the little yellow tag is saying, go to another organelle. In this example, the orange tag is, no, let's ignore that one for now. In this example, the purple tag is saying, go to the plasma membrane. In this example, this orange tag is saying, go to the lysosome. Ugh. Okay, so you can see that each of these various tags acts as a signal to tell these protein products where to go. All right, now let's focus in on the lysosome as an example of how you might ship proteins to another part of the cell to do a job. Okay, so in this example here, let me scroll up. What I'm showing you is an outside of the cell and inside of the cell. And in this example, the cell is taking in some different things. Here we're taking in like a bacteria maybe, through a process called phagocytosis. Here, maybe we're taking in some protein product from another cell, okay? Here, we're actually breaking down an old and tired mitochondria. Remember that the job of the lysosome is to break down cellular products. It's the recycling center of the cell. Now, in each of these examples, what we want to do is to break down these items. Notice that they're surrounded by a cell membrane here that's been pinched in, or in this case, it's just been made. Here it's been pinched in. Now, how are we going to get the protein products that we need to break these things down? Well, basically those protein products are going to come from the Golgi apparatus, okay? Those protein products are usually enzymes. And they're digestive enzymes, so they're going to break stuff down. Okay, so these digestive enzymes come from the Golgi apparatus, which is not pictured here, and they will target this what they call late endosome. Don't worry about that, but this membrane product here, they'll target that. They will have some proteins be inserted in there, and then it's a fully functional lysosome that can break down whatever we give it to recycle these products for the cell, okay? So that's an example of how you might have some proteins from the Golgi apparatus be shipped to an organelle inside the cell as opposed to the cell membrane here, all right? Now let's back up for just a second. I told y'all earlier that the primary issue that we were going to talk about, the primary function we were going to talk about was secretion of proteins moving through this system, okay? But there's one other function that this endomembrane system, which consists of the ER and the Golgi apparatus, there's one more function it has. So it has, first of all, protein secretion. The second thing it does is it makes membrane. And if you think about this, this is going to make absolute sense to you. Okay, let's look at this diagram really quickly one more time. What if we didn't have these ribosomes on here? Then we would not have rough ER anymore, we would have smooth ER, right? So we could go through this process and have these vesicles generated without a protein product in the middle, and then when these vesicles fuse with the plasma membrane, what we've done is just added more membrane to the plasma membrane, okay? So in this way, we can use this system to make membranes as well as proteins. All right? The last cell system that I want to talk to y'all about today is the cytoskeleton, okay? So the function of the cytoskeleton is twofold. All right, the first function of the cytoskeleton is to give support to cells, right? Help keep them round or whatever shape they're in, that kind of thing. But the second function of the cytoskeleton is actually going to have to do with movement. Okay, so we're going to see that the cytoskeleton is made up of proteins. And when those proteins function alone, the cytoskeleton proteins, when they function alone, they are going to provide support. Come on. But if these cytoskeleton proteins, so I'm going to put Cy proteins, if they partner with a second type of protein called a motor protein, then they can help things move within the cell and they can help the cell itself move. Okay, so let's back up for a second and look at this figure. The cytoskeleton consists of 3 different types of protein structures. Okay, so the first type of protein structure you find in the cytoskeleton are these actin filaments. These are the smallest. Think of these as little individual threads within the cell, okay? And you can see it's actually like a strand of 2 beads that are kind of like twisted together, but they make a very thin like rope. Think of this as a rope or a thread, okay, inside the cell. What do these actin filaments do? Well, they help provide structure, they help maintain cell shape. They also help cells move via contraction, okay? And we're going to focus in on that in just a second. The second type of cytoskeleton element is called an intermediate filament. So it's a little bit bigger than actin, okay? Its primary function is just support. We don't really see this involved in movement. This is going to be things like keratins, which make up your hair and your fingernails, okay? And so these are like ropes that are just a little bit bigger. If this is a thread, then maybe this is twine, okay? So think of these as like long proteins, thread-like, twine or rope-like. The third cytoskeletal element is called a microtubule. It is bigger. It is a hollow tube, okay? So it's a bigger hollow tube. Think of this like a, maybe like a garden hose, okay? And it's also going to be functioning in support, and this one can help you with movement. So this one here is going to be support. Come on. Plus movement. This one's going to be just support. And this one's also going to be support plus movement. Okay. All right, let's focus. We're going to focus in on these individually. Let's talk about actin first. So first of all, actin, like I said, it's the little thread-like cytoskeletal elements. It is actually the most abundant protein in your body. You'll often find this just under cell membranes. You'll find actin just underneath cell membranes to help cells maintain their shapes. So you can kind of think of it as like, okay, here's a cell membrane, and maybe this is a cell that we want to be round, so we can like lay down a bunch of actin underneath to kind of support the cell membrane, if that makes sense, okay? The second thing that it does is it also is involved in cell movement. As a matter of fact, this is how your muscles contract, okay? So let's take a look at that. I actually combined these 2 things together. Your functions are up here, okay? So cell movement, cell membrane support, most abundant protein. We got it. All right, so I'll just put right here, and that goes there. So let's look at this closely. Here's our actin filament. And remember how I said that in order for these cytoskeletal elements to provide movement, they had to partner with what we call motor proteins. So myosin is an example of a motor protein. So what happens is myosin can bind to the actin filament right here, okay? And then if we add ATP, it'll lift, bind, and move it again. And then we add ATP, lift, bind, and move it again. And so that can cause things to contract. Let me show you a little video that might help explain this a little bit better. So let me change my screen sharing. All right. All right, let's have a look at how this might work. So here at the bottom, this is our actin. It's a cytoskeletal protein. Up here is a motor protein called myosin. And myosin has some different parts to it. Like this is one part, this is another part. And I don't care about you knowing the parts of myosin. I just want you to know that anything in the blue is myosin. Anything down here in orange is actin. And you can see that actin has binding sites for myosin. Now let's watch what happens when ATP does its thing. Shoot, shoot, shoot. All right, so there's our myosin binding sites. Now along is going to come ATP. ATP is going to bind and give some energy and cause this to pick up. And then the ATP breaks apart, okay? Next thing that's going to happen is this is going to bind downstream and push it forward. And we can repeat this process. Here comes ATP again, causing this to pick up. It's going to create a situation where this binds downstream and pushes this actin forward, okay? So going back to our original PowerPoint. All right, so we have a situation where the combination of the cytoskeleton protein and this motor protein result in movement. Where might we see this movement in the cell? Well, I told you that you might see it in muscle contractions. Oops. Why can't I spell today? All right, a second place you might see it is in what we call cytokinesis, which is the pinching in of cells during cell division. And then a third place you might see it is in something called cytoplasmic streaming. What does that mean? If you are a plant cell, you want to make sure that the cytoplasm is always in motion so you can have all of your like nutrients and reactants always interacting with the entire cell. So actin and myosin can work to move things around the cell, okay? There is a fourth time that you would see actin and myosin working for cell movement, and that would be in things like cell crawling. When you see an amoeba moving along, that amoeboid motion is driven by actin and myosin working together, okay? So that covers our first cytoskeletal element, which is actin, which is that very, very thin cytoskeletal element. Let's talk about the intermediate ones, the ones that are a little bit bigger. Think of them as kind of like a rope. They are called intermediate filaments because they're in between the smallest and the largest of our cytoskeleton elements. And these guys really are involved in support. Or another way of saying that is they're involved in structure. So the big examples of this that I want you to think about are keratin, which is the stuff that your fingers, nails, and hair is made of. Okay, so that's really pretty much what intermediate filaments do. They really work on support. Another thing that would be another example of when you would see intermediate filaments, remember me talking to you about the nuclear lamina? That's also made up of these intermediate filaments, okay? The thing about them is they're tough, tough, tough. They have great strength, okay? So they're very good at protecting cells against mechanical damage. And the third and final type of cytoskeletal element that I want to talk to you guys today, are microtubules, okay? They were the large ones. They looked a little bit like garden hoses. If you were to dissect them, you would see that they're made up of actually two different proteins repeated over and over, alpha tubulin and beta tubulin. And these guys combine to form what's called a dimer, or a two-unit structure. And that just gets wound over and over and over again to make this hollow tube-like structure, okay? So they are made up of alpha and beta tubulin, okay? They are large. Oops, go on. They are hollow. Where they are manufactured are these MTOC regions, which stands for microtubule organizing centers. And believe it or not, y'all have been studying about microtubules and microtubule organizing centers since you were in about the 5th grade and you studied meiosis and mitosis. So let's look right here. This is an example of something called a centrosome. And a centrosome is made up of two little, they almost look like little short pieces of licorice. Like if you took a Twizzler and chopped it up, these are called centrioles. Now that's probably ringing a bell. Let me show you a diagram where you might have seen this before. Here, when you see cells dividing via meiosis or mitosis, you've probably seen examples of these centrioles. Each of these is individually called a centriole. Together, they make what's called a centrosome. And basically, they are manufacturing centers where microtubules can extend outward. So here we have microtubules extending outward. And you guys probably remember from your high school biology that these microtubules can attach to these chromosomes and pull them to opposite ends of the cell. So that should tell you something about the job of microtubules. What might their function be? So their function is really in moving cellular components. So just like if you were drawing a cell that's in the middle of meiosis, and you would have your little centrioles here, and you'd have your chromosomes here, and you'd have microtubules that came along and they would grab these chromosomes and pull them toward opposite ends of the cell. So that's microtubules in action, moving cellular components around. They can also move other organelles around. They actually can form little railroad tracks within the cell. So if this is like a larger cell here, okay, and we're just focusing in on the cytosol, if you could look really closely, you would find these little railroad tracks running all through the cytosol. And what happens is the microtubules like act literally as a road or a track for other organelles to move across. Now remember that I said if we're talking about cytoskeleton and movement, we have to have two things. We have to have the cytoskeletal element, which is our microtubule, along with the motor protein. And the motor protein that's involved in moving things around these little cellular railroad tracks is called kinesin. Kinesin works very similarly to myosin from the example earlier. So basically what happens is you can see here that kinesin has binding sites on the microtubule. If ATP comes along, it causes this guy to pick up, put its foot down here, and then pick up and put its foot down here, and then pick up and put its foot down here. Can you see that here we have something that we're moving on inside the cell? Some sort of vesicle, or maybe it's another organelle. Here we have our little railroad track that's a microtubule, and here the kinesin is acting as the motor protein, where ATP is going to be used to help it pick its feet up and move it, pick up, move, pick up and move, and make its way across the cell where it needs to be, okay? Another place where you find microtubules is in cilia and flagella. Okay, so here's some examples of cilia and flagella. Cilia here are on the outside of this cell. Maybe this is a paramecium or something. And what they do is they kind of wave around. These are like little hairs that can move, and they can move things over the surface of the cell, or they can even help this thing crawl through water. A flagellum is a whip-like structure here, okay? And it functions like a tail that helps to propel a cell through some water, okay? It turns out that cilia and flagella share a similar structure. They are both made up of microtubules. Okay, so I have this word axoneme here. Let me show you what that is. If we were to take a cross section through this flagellum here, and here you can see here's a sperm and that's a flagellum, and we're taking a cross section here. Here's what it would look like. Each of these are a set of microtubules, okay? And they're arranged in doublets. There's 9 different doublets around the outside, and there's a couple in the middle. This whole structure together is called an axoneme. So where you have this set of doublets with the microtubules in the middle, all of these are microtubules. This whole thing together is what is called an axoneme, okay? So this axoneme, which I'm just going to define it for you, it's a set of microtubules. Come on, pen. It's a set of microtubules. You find it in cilia and flagella, and it consists of 9 microtubule doublets with 2 in the center. So you'll see this called a 9x2 arrangement, all right? And you can count that out to satisfy yourself that it's true. And when I say doublets, these are just 2 microtubules that are linked to one another. Can you all see that? Okay, so these cilia and flagella, they move the cells. They either move the cells by kind of waving around like little hairs here, or they whip around here. Now remember, I told you that anytime the cytoskeleton is involved in movement, it needs a partner. And in this case, we're going to have a motor protein partner called dynin, D-Y-N, or no, sorry, I think I said that wrong. Yeah, no, dynin. So dynin works just like the other proteins that we've seen earlier. It is another motor protein. Basically, it has little arms that attach to microtubules, and then as we use ATP, the arm moves forward. And you use ATP and the arm moves forward. And you use ATP and the arm moves forward. And in doing that, you can create movement in your flagella and in your cilia. Okay, so let's insert a new slide. All right, and I'm going to make it, hold on just a second. I want to just do a little review because I think this is a fairly complex lecture. So we talked about cellular transport, or sorry, talked about cellular systems, just as a recap. Oh, come on. All right, sorry y'all, I'm having terrible technical difficulties today. I just wanted to recap. This lecture is all about cell systems. I feel like this is a very information heavy lecture, so let's do a real quick review. We talked about nuclear transport. Right? And so what you need to know about nuclear transport is that the nuclear localization system targets and allows entry into the nucleus, right? Make sure that you understand that, okay? There are similar signals that allow exit too, okay, from the nucleus. Can y'all see how I'm having a hard time? I don't know why come on, come on, come on. Okay, so similar signals. We also had the endomembrane system. The endomembrane system was made up of the endoplasmic reticulum and the Golgi apparatus. It had two jobs. It could make proteins and it could make membranes. Where these proteins or these membranes go were also determined by various signals that could be chemical, like a protein, okay? It could be sugar or whatever, but I showed you guys that diagram where you could see that the different color tags were sending these little vesicles to different regions of the cell. And then the last thing that I told you all about was the cytoskeleton. Cytoskeleton, all right? And I told you all that it was made up of 3 different types of proteins. We had actin. We had something called intermediate filaments, which are actually a series of different protein types, but they make up like a rope, intermediate filaments. And we had microtubules. Right? These can function, the actin filaments can function in support. Remember I said that they would often like surround cell membranes. They can also help in cellular movement. Okay, these were involved in structure. Oh, lordy, lordy. And these were also involved in movement. And remember that anytime you have the cytoskeleton involved in movement, you need the cytoskeletal protein plus a motor protein. And the 3 examples of motor proteins that I gave you were myosin. Oh, Lord have mercy. Myosin, kinesin, and dynin. Okay. The one that I really want you guys to focus on is understanding how myosin and actin work together. You will have that again if you take physiology or take other biology courses. These, I want you to understand how they work. I don't care if you understand their names, okay? So that might help you outline your studies just a little bit. If you have any questions about this lecture, we can talk about them on Monday. Bye-bye.